Hi everyone. I know that you're doing well and your studies are going good, but just let me tell you one thing. You people are in class ninth. Once you will in class 10th and then you will be in 11th. My point is your ninth and 10th, you should have 100% grip over all the topics. Just learn carefully and just because these are basics things. So learn in a proper way so that you don't have to struggle in your 11th and 12th just for clearing the basics. So just learn it up in a good way. Now we are going to start the topic. Okay, so up to now into the cell chapter, we have covered it up to all the organelles. Now, the very basic things they are left in the chapter that we are going to do the differences and one or two organelles that I'm going to mention. So, for that, I have to share my screen and let's try. Okay, so it's here. I suppose you can see it. So basically, let me just tell you in a very brief way what we are going to discuss. As I was saying, we are going to learn the differences. Prokaryotic and eukaryotic cell, plant cell and animal cell. I guarantee you in a paper from the last part that I see is very easy, but you can have at least 3 marks of question in your board exam or also in your Olympiad. One question at least in Olympiad. So the differences, they're really, very important. Seriously, I'm telling you. Cilia and flagella, centrosomes, and a little bit about the cell division. Okay. So the first thing that we have to cover is differences between prokaryotic and eukaryotic cell. So my point is that first of all, you have to understand what is the idea. I always tell it to my kids, my all the students, that bio is not English. It's a different language that you're learning with the help of English. It has different terminology. The most important thing to learn about bio is that you just need to crack the words. Once you crack the words, you get the idea, you get the meaning. Okay, like suppose pro means primitive, purana, old. And wherever you see the word karyo, that means they're talking about the nucleus. U means true. And then karyo is nucleus. So my point is, prokaryotic is an old type of cell which is not having a proper, well-established organelle and everything in the cell. But eukaryote, this is a true cell, which is like a well-established. It has everything in it. Now, if you look at the structure, the very basic structure is bacteria itself. This bacteria structure. Now, guys, into the bacteria, if you see, it's having some basic organelle. Cell wall, cell covering, cell membrane, everything. You've learned that in your 8th class also. So plasma membrane, cell wall is there. And then inside genetic material. If you see genetic material at the center, which we suppose is nucleus, but we do not call it as nucleus, we call it as nucleoid. Because it's just a simple genetic material which is not even covered by the membrane. That's a basic difference between prokaryotic and eukaryotic cell. So this is not covered up by any membrane. And I have taught you a lot of organelles till now. Prokaryotic cell do not have all the organelles. It has just a little few, like ribosomes. Yeah, I showed you a table already. I'm going to mention all the difference, but this is the very basic picture. Yeah, they have flagella also. I'm going to teach you cilia and flagella also. But in short, understand it for now that they are the locomotory organs. They help in locomotion. Now, talking about eukaryotic cell, plant cell and animal cell, they are eukaryotic. Even fungi, eukaryotic. Protista, eukaryotic. Kingdom, actually, if you have learned diversity chapter, na, there's a whole kingdom which is known as Monera. All monerans are prokaryotic. And then rest of, there are 5 kingdoms. Monera, Protista, fungi, plant and animal. In the life, on the universe, if I talk about all the animals, all the life, I would say. So Monera has all the prokaryote and rest of fungi, protista, plant and animal, they are eukaryotic one. They have all of the organelle, a proper nucleus, membrane, genetic material well established, all the organelles properly present, doing all the work. There's a well distribution of work among them. Getting my idea? I have a video also I'll show you. But before that, let's see this table for the difference. You should learn that important. Okay, so eukaryotic cell here, nucleus is present. Here it is absent. Actually, nucleus means a proper nucleus which is covered by the membrane. So you can say enveloped nucleus. Enveloped means which is having the membrane on it. Number of chromosome more than one here, one, but not true chromosome. Actually, DNA is made up of chromosome. In eukaryotic cell, you will find obviously we do are having a human is having 46 chromosome, means 23 pairs, so more than one. But in the prokaryotic one, just one DNA, coiled and coiled and coiled. That's it, and no true chromosome. That's what. Cell type, usually multicellular, and these are usually unicellular because mononeurons, all mononeurons are unicellular. Okay, so I'm going to give you example of mononeurons also, usually unicellular. Some cyanobacteria may be, actually I will not say the cyanobacteria is multicellular itself. They used to be present like a colony. Many of the unicellular cells are present like, in a group. We call it as an aggregate or a colony, but they are unicellular. True membrane, that's what. Example, animals and plant of the eukaryotic cell, and here bacteria and archaea. Other than this, there are many unicellular organisms under the Monera kingdom. For example, cyanobacteria, mycobacteria, it's bacteria, and mycoplasma. Yeah, so these are all the examples. I suppose that's clear to you. Let's move further. Genetic recombination. Before I proceed with this point, I'm going to put this point on hold because I'm going to give you a little description about the cell division in this part. And there I will mention genetic recombination. For you now, you just remember that here into the eukaryotic cell, genetic recombination happen, and in prokaryotic cell, doesn't happen completely. How? I'll come back to this presentation. Fine? This slide, sorry. I'll come back to this slide once I'll be from there. I'll be moving here. Okay. So lysosome here present and here absent. So with this, I suppose everything is clear to you, but you need to have a look at the prokaryotic and eukaryotic cell. Let me just share my screen and show you that video. Okay, so there is a new share. That's my favorite video. I show it to all the students. It's animation I like actually. I'm going to put this on mute and I'll speak. You have to listen to me. Okay, so what in this video, let me just zoom this video a little. So into this video, they are showing a cell, that the cell, eukaryotic cell, it's having all the organelles. It is having cytoplasm, it's having the nucleus where its DNA is present, and all the organelles. See how beautifully they are present. So this is a broader category I'm telling you. The bigger one, it's having all the cell organelles and everything in it. This is unicellular and not unicellular, I should say for now, but this is a prokaryotic. I take my words back. This is a eukaryotic cell which has all the well-established organelle. But the little one that you see on the right side, this is prokaryotic cell. DNA is the singly present in the this prokaryotic cell, and into the prokaryotic cell, bacteria is the typical example. Now in this video, they are trying to show all the organelles. If you see the video now, see, it's having the nucleus. You know, this brown or dark red thing that you see, these are actually the mitochondria. Seem like nuts. Okay, so this is prokaryotic cell. Genetic material is present just like that. Not too much of the organelle is present, but cell membrane is there, and inside ribosome is present. I'm going to tell you what things are present. Bacteria is the typical example. What are organelles? I have taught you all the organelles in the last part. But if you want to see the video, I'm going to give a quick view. This is DNA. This is DNA present inside the nucleus. This is ribosome released from the nucleus, coming outside from the pores. I should have shown this video yesterday itself. I mean, in the last part. But that's fine. You're watching it now. See, so point is that from the nucleus, this yellow thing is the ribosome coming outside and joining the endoplasmic reticulum. And also in the cytoplasm, it is moving. See, rough endoplasmic and the smooth one will be the smooth. Proteins are formed from the endoplasmic reticulum. Proteins are coming in the vesicles. You see, from the vesicle, how it is getting released. And then it will go toward the Golgi apparatus. Now they're going toward the Golgi apparatus. Here, packaging will be done. Cis phase and trans. They are taking entry from the cis phase, and from the trans, they will get out. And internally, they are getting matured. They're having a proper shape they should have. See, all the protein have their particular shape. They just get the shape here. How they get the shape? By making bonds, the chemical bond they make. This is lysosome. I have shown you this GIF file, na? Lysosome. This is mitochondria. Inside the mitochondria, how energy is getting formed. This is internal membrane. Na? That's a beautiful video. It's a microfilament. Microfilament, actually, these are skeletal part of the cells. In the cell, that is a skeletal part. And into the plant, they have shown chloroplast. How beautiful this is. Into this is plant cell, and inside it, chloroplast is there. And a big one is the vacuole. Then they are showing the alveoli. Actually, the cilia flagella they are showing. The cilia flagella today in this part, I have to teach you. See how beautiful. Nice. I will show you this. I have some GIF files in the presentation itself. Let's come back to the presentation thing that I was showing you. Here. I suppose everything is clear to you regarding prokaryotic and eukaryotic cell. The only point that you did not understand for now, I don't know whether you understood, but I'm going to put it on hold, the genetic recombination part. Once I talk about the cell division, I'll come back here, or probably you can read it. I'll mention it again. Okay, so let's proceed. The next one, difference between plant cell and animal cell. Plant. They are not wearing any winter clothes or something like that. Doesn't matter whatever the temperature be. That's why each and every cell is protected by cell wall. In animals, that doesn't happen. We do not have the cell wall, but in our cells, we have only plasma membrane. So now talking about the plant cell, usually they are larger than animal cells, and animal cell, these are usually smaller than plant cell. If I talk about the size, because plant cell is having proper junction between the cell, two cells are connected, middle lamella, cell wall, plasmodesmata is one thing which is connecting the 2 points. I think this is written here. Cell wall is present in addition to the plasma membrane. I have mentioned it, and consists of middle lamella, primary and secondary wall. Though I mentioned it, primary and secondary wall in the previous part, the primary is initially that is formed, and secondary later on after getting mature. Here, cell wall absent. Plasmodesmata. Like suppose these are two plant cell, there is a junction, or these are, you just say junction, which helps in transport of material across the two cells. That is known as plasmodesmata in the plant cell that is present, but in animal cell, it is not present. In animal cell, junctions are of different type. They are present, but that happen in a different way. Here, chloroplast is present here. Chloroplast is absent. Vacuole, I have told you this point. Tonoplast present around and tonoplast absent. Yeah, I forgot. I did not mention at that time. Yeah. So the membrane of the vacuole is known as tonoplast, but here, and not as such any membrane is present. It's just a bubble kind of thing which is present, like contractile vacuole or phagosome. Temporary. Tonoplast present around, central absent except motile cell of lower plant, and here central present. I have not mentioned this point. I'm going to mention it in this presentation only, centriole, but you understand this point only, that centriole, it helps in cell division. And there used to be two centriole present in any cell. In the animal cell I'm talking about. One centriole, like suppose two centriole is present, and during the time of the division, one centriole move here and one centriole is there. And then they attract the chromosome from the center. And that's how chromosomes are separated in animal cell. In plant cell, not centriole is present, but cell division also happen. But that protein is present of which centriole is made up of. That protein is present that itself helps in cell division. Same phenomena happen in them, but not as such centriole. Centriole is also made up of certain protein. Okay. So I hope you're getting this point. In plant cell, centriole is not there except lower plant, and here this is present. Nucleus present along the periphery. I have told you this point also. Lysosomes are there, and lysosomes are present. Storage material is starch grain, and here glycogen. Starch is present, amyloplast, leucoplast may. I have told you amyloplast. You can mention it in the difference. And here the glycogen is actually, this is stored in liver. What happens when we eat a lot of glucose and our body, you know that insulin is the one that controls glucose level of the body. That insulin actually converts glucose into the glycogen, and that is stored in the liver. Everything clear? Now, I know that central part you might still be confused with, but do not worry. The next part, I guess, no, next is central itself. I'll come back to this part. So I suppose the difference between plant cell and animal cell is clear to you. These are 10 points I have told you. If that can come as a 5 marks question, I know that you will be able to write, right? Okay. Okay, so the next part that we have to cover is cilia and flagella. The first thing I have mentioned it earlier also. Cilia and flagella, they are the locomotory organs. They helps in locomotion. Organelles, sorry. I was thinking, मैं कुछ तो भूल गई. I did not forget anything. I'm just writing. So basically, basic structure is same. They're made up of proteins. Flagellum, basic structure is same. But the location they are present is different. And even function is also same. So if you see cilia, it used to be present in high number, like hundreds or even more than 100, 200, 1,000 itself into the cell, and it helps and beats like, synchronized movement like this, like beating and beating, and that's how they help in movement. And even cilia also entrap the waste particles, the waste as in pathogen. Like cilia is present in our trachea, it is present in our nostrils, in the cellular level. They also entrap the dust particle also wherever they are present. Like if I talk about, if you have learned reproduction chapter in 8th class, into the female reproductive system, fallopian tube ke andar, into the fallopian tube, ciliated epithelium is there. There it help in movement of the gametes. So cilia is kind of helping in the movement, not itself, but other things also, which is nearby. Okay, so flagella is always present in less number, 1, 2, 3, not more than that. 4, 5 itself, but not more than that. So flagella is okay. I did not see that written thing is also there. Cilia are slender protuberances, as in protuberances means an extension kind of thing that project from much larger cell body. A flagella is a lash-like appendage that protrude from the cell body, and it is present in less in number. Now I'm going to give you example. I suppose it's written there. It is. Though I have given you example already. Cilia is present in paramecium, ciliated epithelium. Okay, it's present in, okay, it's present. They have written simple example. I think these examples are much enough. If something strike me up, ciliated epithelium, if I talk, it is present in the nephron, in kidney, in trachea. That will be just location. Yeah, you can write location itself. That's fine. By the way, next chapter is tissue and cilia. I think you will learn there. Ciliated epithelium, it is in your syllabus, and you'll be learning that in next chapter. But for now, I have told you 2-3 points. Remember it. Cilia is comparatively more, I mean the number, and flagella is comparatively less, 1 to 8. Yeah. Acha. Okay. Since it has the GIF file. Cilia is usually shorter in length, and I have told you this is longer in length. Beating pattern of cilia is very complicated, can move in a wide range of motion, and beating pattern is actually flagella wave-like, propeller-like, but cilia also move like this, but different cilia present at different locations. They can show a little difference, but they also used to move in a synchronized manner. Actually, if I say cilia is more synchronized as compared to flagella, flagella is not synchronized because flagella works as a separate. It shows circular motion, it shows wavy motion. That thing is written right, but not synchronized. But in cilia, this motion is synchronized. This is present in eukaryotic cell. I have told you the example, and this is mainly present in prokaryotic cell, like a sperm. Sperm is not prokaryotic, but sperm also has a flagella. Mainly, it's in prokaryotic cell, and obviously in eukaryotic, you can see. Acha, 1 minute, I'll play the GIF file and I'll show you. This is present in bacteria, prokaryotic cell, and in eukaryotic cell, a sperm example I have shown you. Hold on, now you see. So the last part, the tail part, is actually flagellated. In bacteria, they have shown. This is flagella up there, and this is cilia, present in high number. I suppose that is clear to you now. Now let's move. So the next thing is centriole itself. You were getting confused at that point. That can come in your Olympiad paper. Many students get confused at this point. Let me explain you. Basically, the thing is centrosome. I told you centriole is present always 2 in number. In the cell, there are, like suppose this is a cell, it'll be 2. If we magnify it up, it look like this. Like the wires entangled together a lot of, and then they are present at the 90 angle, perpendicular to each other. Together they are known as centrosome, and separately this is centriole. 2 centriole make 1 centrosome, remember it. Now, into the cell, during the time of cell division, 1 centriole move here from the centrosome and 1 here. And then from the center, nucleus are present, and they are actually the chromosome present, and they attract the chromosome. Half the chromosome attracted here and half here. That's the reason that chromosomes are separated from each other. And this is made up of, now I tell you one thing. This one centriole, it is having wires like this. These wires are known as microtubule. So a lot of microtubule make one centriole, then two centriole makes centrosomes. Remember it. Microtubule is further made up of protein. I have told you, tubulin protein is there. Other proteins are also there. For now, you just remember this name. I repeat myself. This is microtubule. Together, they make centriole, and then two centriole together make the centrosome. Now I'll come back to that plant and cell, this difference. Central absent except motile cell of lower and inner central present. In animal cell, central present, a proper cell division happen. But in plant cell, central as such is not there, but microtubule thing is there that helps in the cell division. I hope that's clear to you. Let's move further. Okay. So the next point. Okay, I have to tell you cell division only. Do I have the space? Let me see further. Question answer. Okay. Okay, the space is enough for me. So basically, cell division. First of all, the phenomena of the cell division, it was explained by Rudolf Virchow. You can write it in the history. I have told you in the cell theory that he told that omnicellular is cellular, means all cells arise from the pre-existing cells. That's what he was trying to say, that cell divide. Now, talking further. Cell division happen in all living creature, mainly in plant and animal cells, centrioles, and the microtubule helps in cell division. I have told you the brief out. Now let's understand cell division types. But before that, let me tell you one more thing. Whenever a cell divide, first of all, its nucleus divide. First of all, everything inside the cell become doubled. DNA, organelles, everything, it become doubled. And then cell nucleus divide. First of all, nucleus will divide and then the cytoplasm divide. So you may say there happen two steps. One is karyokinesis and one is cytokinesis. I have told you this before, but I did not change the pen color today. I have told you this earlier also that karyokinesis is karyo, what is used for the nucleus, and cyto, this is used for the cytoplasm. And kinesis means splitting. So first splitting of nucleus happen, that is known as karyokinesis, and then cytoplasm divides into half, and that is known as cytokinesis. These are just the steps. But let's talk about the type of cell division. First of all, cell division used to be of two types: mitosis. In general, I am talking about in all the cell, in meiosis. One thing that you have to remember, mitosis always used to happen in the somatic cell, and meiosis, it also always used to happen in the gametic cell. Soma. So wherever you see the word soma, that means body. Somatic cells means body cells. Like all the cells in our body, they used to divide by mitosis. We get a little injury, we get hit, a little injury is there, so cells will be dividing to heal it up. Mitosis is happening. In the gametes, in the testes and ovary, in plant, into the pollen, into the flower, ovary and pollen is there. These are the places where gametes are found. There meiosis happens. So meiosis used to happen in the gametic cells: sperm, ova, pollen, and the plant gamete, female gamete. Clear? Now, let me tell you one thing. Mitosis, it used to be equational division. And meiosis, it used to be the reductional division. Now, let's understand the meaning of it. Equational means chromosome number remains same. Like suppose any cell is having 2n chromosome, it will be 2n only after the division. 2n means double pair. I will tell you that thing also. And here suppose a cell is having 2n before division, it will be n after the division. The chromosome number become half. That's why we say it's a reductional division. 2n and n is actually the pair of chromosome. The n word, if you see, that means a set of chromosome. Chromosome. Okay. So n is the set of chromosome. 2n means double set of chromosome and n means single set of chromosome. Like human used to have 46 chromosome, 23 pairs. The value of n is 23. So two set are present. Gametes used to have N, only 23. One sec. Is that clear? Are you getting my point now? When sperm is formed and ova is formed, this is S. They used to be N and N. And while the time they divide, they become 2N again. And then while 2N, they start dividing. And then a human is formed. Are you getting my point now? This is mitosis and this is meiosis. I have a video also on the mitosis and meiosis, the detailed one. I think for now, this information is much enough for you. But if you want to go into the detail, I have added that in your notes. If you want, you can contact. And let me show you that video in very brief for now. In this video now they have shown like how chromosomes are getting separated. Actually now when chromosome used to get separate from each other, it used to happen in certain stages. Chromosomes is getting hard and then it's moving up, and so we have divided it in different stages. Like they are trying to show into the mitosis. I will tell you one more thing. Into the mitosis, see, they are saying that chromosome is there. See, into the cell, centrole is moving apart, and into the chromosome, into the center nucleus, chromosome is getting hard. It's getting a proper shape, and the centrole is moving apart. And then this starts to get half and half. First of all, they will move apart. They will form some fiber kind of thing because these fiber only will attract the chromosome. This fiber only will be holding up onto the chromosome. These fiber move apart, and then they, take the half of chromosome. See how chromosomes are getting separate. And then these fiber thing, which we call it as spindle fiber, they get disintegrate, means gayab ho jate hain. They just disappear. See how chromosomes are getting half and half. Go! Why you stopped chromosome? Okay, they are going. See how they are moving. Now everything will become disappear. And both of the part will have its separate membrane. I'm going to tell you one more thing, guys. See how they're moving. I want to tell one more thing. Even if it is mitosis, even if it is meiosis, even if it is mitosis, even if it is meiosis, before mitosis and meiosis, there is a phase which is known as interphase that happened. Interphase means when cells content is getting doubled. DNA, everything is getting doubled. That is interphase. And then when cells start dividing, we call it as M phase. In mitosis, we call it as mitosis. In gametic cell, we call it as meiosis. But interphase happen in all the stages. Clear? So I suppose now you have a clear idea about the cell division. And I suppose everything is clear. But if there is a doubt, watch this video again. Move backward, watch it again, and then, acha, sorry, I forgot to mention one thing. I was about to say you bye, but then I was telling you one point, genetic recombination wala. I told you after cell division, I will tell you. Thank God. Yaad aa gaya. Genetic recombination. We say that we look like our parents, but we are not exact copy. Some genetic recombination means some genetic changes happen in us. That happen when sperm and ova is getting formed. Because sperm and ova react together, then a baby is formed. But before that thing, when sperm and ova is getting formed, at that time, genetic recombination, some changes happen. We call it as genetic recombination. So now into the eukaryotic cell, meiosis and fusion of gamete happen. Eukaryotic plant and animal are eukaryotic. They show meiosis in their gamete. They have fusion of gamete. And that's the reason that new combinations are formed. Male combination, female combination, into the meiosis itself, some combination. But in prokaryotic cell, no such phenomena, partial unidirectional transfer of DNA. One cell will be connected to another, a tube-like structure will be formed, and DNA will be copied into the next DNA. That's how it happened, into the DNA. They don't as such just show the phenomena that I just explained. And now I finally suppose everything is clear to you. Let me come back. There was a question also, no, which type of cell division is required for growth and repair? Mitosis and meiosis. I don't think an explanation is needed. Thank you. Thank you. So I suppose everything is now finally clear. And if you have any doubt, just put it on the forum where you know that clear, where you have to put it and where you have to ask. So bye-bye.